The overall goal of this experiment is to analyze the motor neuron projection patterns in late-stage embryos of Drosophila melanogaster.
This method can help answer key questions in the field of neural development, such as motor axon guidance and neural circuit formation. The main advantage of this technique is that it provides precise visualization of motor axon in developing embryos, which allows for reliable analysis of axonal pathfinding and target recognition defects.
One day after setting up egg collection plates, replace the plates with new ones containing a dab of freshly made yeast paste. After 3 hours on the fresh plates, transfer the flies to new food bottles. And incubate the egg plates overnight at 25 degrees Celsius. In the morning, add 1.8 milliliters of PBT solution to the plates. And use a cotton swab to dislodge the embryos.